So hello and welcome to the second episode of the BrainFacts Brain Beat Prep tutorials held by the Central Ohio Youth Neuroscience Coalition. Today we'll be talking about the first topic in Chapter 2, Senses and Perception, which is about vision. So first of all, let's talk about some general information about vision. First of all, the first thing you should know is that vision takes up an extremely large amount of processing power. It takes up about 30% of the cerebral cortex, and it has an entire lobe, which is the occipital lobe, dedicated to processing it. And then through research, we've learned a lot about vision. So specifically through mice and Drosophila, which is basically fruit flies, we've learned about the knowledge of light to an electricity conversion. So basically, we've learned how the brain translates light signals, which come in through your eye, into electrical signals the brain can process. And then we've also learned about higher level processing, so like how do you detect contrast, color, all of that kind of stuff through research done in monkeys and cats. And then the overall process, which we'll be talking about a lot more later, is it goes from the cornea to the pupil to the lens to the retina to the optic nerve to the optic chiasm to the optic tract to the lateral genicular nucleus of the thalamus, and then finally to the primary visual cortex and the occipital lobe. But we'll be talking about this more in depth now. So now let's talk about the first step of the process, which are the pre-retina processing. So first, light hits the cornea, which is basically on the very outside of the eye, and then light enters eye through the pupil. So the pupil actually affects how much light is let in, and the pupil size is controlled by the iris. The iris controls the pupil, which controls how much light is let in. And then after that, the lens focuses the image onto the retina. And so the cornea does a lot of the primitive focusing, but the lens does the most focusing. So always, if you're asked a question about the focusing, say the lens, not the cornea, because it only does primitive focusing. And then the lens, the way it does the focusing is that it thickens and flattens itself. And then also, one other thing to remember is that the things where image in the real world is actually reflected on the retina. So things on the left are on the right in the retina, and things on the right in the real world are on the left in the retina. So this is probably the most important part of this chapter, which talks about the retina. And so if you're gonna go and study for the brain bee, I would recommend that you focus on this because there's a lot of stuff that you need to know about the retina. So basically the retina makes up the inner surface of the eye, and it has three different types of neurons. So there's photoreceptors, interneurons, and retinal ganglion cells, or RGCs. So we'll talk about them all, and then we'll have like a diagram where we explain the different how the signals pass through. So first of all, let's talk about interneurons. Interneurons are basically an intermediate between photoreceptors and RGCs. We don't usually talk about them much. They're not like in the BrainFacts book very much. So if you go into like a higher neuroscience class, you might learn about them, but like we won't talk about them here. And then photoreceptors are very, very important. They convert the visual light signal into an electrical signal. And so there's two types of photoreceptors. There are rods, which are important for dim lighting. They are seen, they're actually way more common than cones. They're like 95% of the photoreceptors in the eye. And so they're important for dim lighting, but they can't see anything like visual acuity or color. And they're also really important for like peripheral vision. And then there's also another type called cones, and these are really important for visual acuity and color. And then there's three types of cones, R, G, and B, so red, green, and blue. And so those cones combine they combine in the brain, their signals, and so it forms all of the colors that we can see. So in the retina, the highest concentration of only red and green cones, remember, no blue, only red and green, is located at the very, very center of the eye. This is called the fovea, and then this is where the highest visual acuity is. So you can see the best in the center of both of your eyes. So this probably makes sense because, I mean, if you want to focus on something, you look at it with the very center of your eye, right? And then this is because, of course, the large amount of cone cells, the cells that are important for acuity. And then the area immediately surrounding the fovea is called the macula. This is important for reading and driving. If you've ever heard of macular degeneration, which I'll just write right here, macular degeneration. This is a disorder that's seen in older people, and where they lose the function or they lose the photoreceptors within the macula, so they're not as good as at some of these things. Now let's talk about the last type of cells that are in the retina, which are called the RGCs, retinal ganglion cells. So these cells are really, really special. They have this special system of processing called the center surround system. So basically, if this is like an RGC, and then this is its center and this is its surround. If it's hit in the middle, it's excited, so it wants to send more signals. If it hits on the outside, it's inhibited, so it wants to send less signals, and then if it's hit in both parts, then it's only weakly excited, so it responds but not as well as it would before. So this actually emphasizes contrast, which is a big thing in like vision in general, in vision processing. Contrast is really heavily emphasized and like it because it's really important. So you want to know the difference between an object and its surroundings. So it's really heavily emphasized through the center surround system of processing. So now that we've looked at more of like the basics of the retina, let's try to diagram it out and try to draw all of the retina. So right now I'm just drawing the retinal layers. So we have the photoreceptors. Oh, why did I say PC? We have the photoreceptors, and here we have the interneurons, and then here we have the retinal ganglion cells. So basically, there's like three different layers, and what you might be surprised to find is that the front of the eye is actually located all the way over here. So it's actually pretty far from the photoreceptors, and the back of the eye near the brain is actually located all the way over here. So what happens is that it goes over here. Well, it doesn't really go from there, but like because usually these layers aren't exactly like this, but what I mean to say is that just that it goes from the front of the eye to the photoreceptors, and the photoreceptor signals go to the interneurons, and the interneuron signals go to the retinal ganglion cells, and the retinal ganglion cells, their axons go all the way to the back of the eye. So again, it's not like over here. It doesn't really cross across the side, but I'm just doing that for simplicity purposes. So the retinal ganglion cells, these axons form the optic nerve, which you might have heard of because it's a cranial nerve. Basically, it takes the signal from the eye all the way to the brain. And so where these retinal ganglion cells cross the eye, there is a thing called a blind spot because you can't see anything where they're crossing because they're crossing, of course. Their axons are crossing. And so that is just basically like a simple diagram of how the retina works. There's a lot more layers and steps involved, but just let's keep it simple here because Brain Bee does not want you to know everything about the retina. And so now let's look at the fovea and the macula. So basically what happens is that say this is the retina, like this whole thing. Then in the very middle, this is the fovea. And then at the sides, these are the macula. And then all the way over here would be like peripheral vision. And so what happens is that this is a PR layer, right? This is the photoreceptor layer of the retina. And then so you would have, theoretically, you'd have like your interneuron layer, and you would have your RGC layer. So what happens is that if you have a neuron in the fovea, it sends a very each or what I mean to say is that each retinal ganglion cell has a very small amount of foveal inputs. So like if you had a cell right here, an RGC right here, then it would maybe only have this from 1 or 2. Whereas like for a peripheral cell or for somewhere that's a little bit farther, it can have like a lot of inputs, like 4 or 5 or something like that. So this is also another reason why there's such a high visual acuity in the very center of your eye because because or at at the fovea because the fovea only sends a couple of inputs to each RGC, whereas in the peripheral, in the more peripheral parts, each photoreceptor sends a lot of, like each RGC gets a lot of inputs, if you see what I mean. So now let's talk about after the retina, but also before the visual cortex. So after exiting the retina at the blind spot, we just talked about the fibers of the optic nerve or the axons of the RGCs for each eye go to the optic chiasm, where some of them cross over, and then basically once they cross over, they go to the optic tract. So essentially what happens, and this is going to be a little bit confusing at first, we'll explain it in a diagram right after this, so that might be a little bit less confusing, is in the left optic nerve. So before is you have the left half of the left eye and the right half of the left eye. So you have all of the left eye. But after, in the optic tract, you have the left half of the left eye and the left half of the right eye. So some of the fibers cross over. And then in the right optic nerve, basically the same thing happens. Usually you have like from the eye in the before the eye chiasm, you have the left half of the right eye and the right half of the right eye. So you have both halves of the right eye. But then in the right optic tract, I'm not sure why this says left, in the right optic tract, you have the right half of the left eye and the right half of the right eye. So let's talk about this a little bit more with a diagram. So essentially what happens is that if this is the optic chiasm, like this, it kind of looks like this on an anatomy model, but not that great of a drawing. So what happens is that this would be like the, let's just call this, oh, this. All right, let's just call this the optic tracts would be over here, and then the optic nerves would be over here, right? So over here, this would be the right optic nerve, and this would be the left optic nerve. And then after this, this would be the left optic tract and the right optic tract. So what happens here is that from the left optic nerve, you have some fibers that decide to cross over into the right optic tract, and then from the right optic nerve, you have some fibers that need to cross over into the left optic tract. And so it's crossing over. It's an optic chiasm. And so yeah. So now let's talk about the brain pathways that are involved. The optic tract basically leads to this place called the lateral geniculate nucleus, or LGN, of the thalamus, which is a pretty big vocab word. You should probably know that. And then this leads to the primary visual cortex, which is also called as V1 in the occipital lobe, so which is at the very back of the brain. So basically it goes from like, if this is a brain, it goes from like where the thalamus is, so like right here-ish, to like all the way back here. And then this is of course where the eye is. So it has a pretty big path. And then like just in general, the eye pathway, the vision pathway has a pretty big path. And so V1 has many, many layers. The lateral geniculate nucleus actually feeds into the very middle one. And in the middle one, it produces the exact same image as was on the retina. So, but otherwise, so like if you had these types of layers and you had V1, V1, this would be the middle one, and the lateral geniculate nucleus would go all the way just into here. And then after that, they would go either over there or over there, depending on like their process, or maybe they'd go over here and then over there, or over there and then over here. So it all depends on what type of info it is. So obviously the upper and lower layers would have more complex fields, so they have a lot more filters and processing and stuff like that. And so these are also these are parallel and interacting systems, all of these fields and these filters and like the layers. So basically what happens is that one of these, or a couple of these layers are known as, or a couple of these systems are known as the dorsal system and the ventral system. So the dorsal system, the information the information output from the dorsal system goes to the parietal lobe. So basically the parietal lobe deals with like touch and feel and stuff like that. So it's known as like a what sensation, whereas the ventral system goes to the temporal lobe. So that's like associated with hearing and memory and stuff like that, and like spatial awareness especially. So it goes and looks at the where aspect. So there's a lot of different aspects of vision as well that come into play. Lastly, let's talk a little bit about binocular vision. So this will be like the very last thing that we cover before we go into diseases. Binocular vision is basically vision with two eyes. This vision lets you perceive depth and dimension, which are really, really important. And this happens because each eye sees from a slightly different angle, so it lets you see the depth of any objects. And so for this to happen though, the visual fields have to overlap and then the eyes must be aligned for this to happen. So in this disease called strabismus, you see that the eyes aren't aligned, so they cannot perceive depth well. And then in this disease, strabismus, basically the eyes are not aligned. So although like kids who are born, they have use of like both of their eyes, they're both working. They just favor one and they neglect the other like automatically just because they don't align. So it's really confusing to see the world with non-aligned eyes. And so this disease needs to be treated very, very early in order to completely fix the problem. I think it's like before you're like 8 or so like that or something like that. And so this has to be done during the critical period, which is basically this big development period of visual development. So before, doctors actually used to wait until like around like 4 or 5 to start treating kids with these diseases, to start treating kids with this. But we found in research using cats and monkeys that waiting is just not a good idea. It has a lower chance of working. And now lastly, let's talk about diseases in which you have a loss of function or death of photoreceptors. So these diseases are pretty hard to treat because you can't grow back photoreceptors. That just doesn't happen. But right now, we're looking at a type of potential treatment where you're trying to send electrical signals to the brain via the retinal ganglion cells and just bypassing the photoreceptors altogether. So you don't need to make them grow again. You just need to develop a device that like looks and interprets the world for you, which is probably pretty hard as well. So but however, researchers are also working on many other treatments, especially for like genetic blindness. And then this is like you can see the cause of genetic blindness through like genetic studies and model organism studies. And so that concludes this episode. So yeah, stay tuned.